All right, last sections for chapter 6, enzyme. Let's start with enzymes. So we talked about enzyme about multiple occasions before, but let's formally define it. So a catalyst is any substance that helps with chemical reactions to occur. So to speed it up, to make it happen, that's what we call a catalyst. Enzymes are actually biological catalysts. So enzymes are catalysts that works within our bodies that will speed up chemical reactions or will catalyze biochemical reactions by lowering the activation energy. So as you can see here, normally this is our reactant, this is our product. Normally it would need to go up all the way uphill like that to have that activation energy and to cross that threshold in order to become a product. So that's really high. Enzyme comes into these pictures and cause this activation energy to lower. You see this lowering energy right here, and it is easier for the reaction to occur if the activation energy is lower. This enzyme makes reactions to occur faster and increase reaction rate. However, that does not change the delta G or the Gibbs free energy of the reactions. As you can see here, this Gibbs free energy stay the same, and it doesn't change whether that become an endergonic reaction or an exergonic reactions. Those kind of reactions will stay the same. The Gibbs free energy release is still the same, or release or absorb is still the same, but it will change the activation energies. Almost all enzymes are proteins. You have some exceptions, enzyme being an RNA molecules, we call that a ribozymes, but most of our enzymes that we'll concern up until our DNA chapters will be proteins. Enzyme has to work with a substrate. Substrate is just a fancier term to say all the reactants. So, but there's some more caveat to that in terms that it has to be very specific. Substrate, there's a specific substrate for each of the enzyme to catalyze a specific reactions. So you can have one or more than one substrate. So you can see here, one substrate break down into two products. You have two substrate combined to form one product, or two substrate go through the reactions to become two completely different products at the end. And all of this with the help of an enzymes. Like we talked about before, protein is very sensitive to pH and temperatures. Enzymes are protein, thus they are sensitive to pH and they are sensitive to temperatures. At extreme pH, or just either too acidic or too basics, the amino acid residue, or that's basically referring to that R group, will actually be deprotonated or protonated. What that means is they will lose an H or they will gain an H, or hydrogen ions. They will gain that and change form, and when amino acid residue change form, it can cause the protein to change conformations. Thus, the sensitivity to pH change makes enzyme cannot function well outside of their comfortable pH, and usually enzyme exists at around our body pH, which is around 7.4. So extreme pH, too low or too high of a pH will cause enzyme to denature. For extreme temperatures, it can alter the chemical bonds. As we can see, temperatures can cause a variety of things occurs. As an example, with our eggs, if we fry an egg, we use temperatures to denature the protein. Thus, the protein element turn from translucent to more of an opaque color, as you can see of frying eggs. Then, that's the overview about enzyme. Now we will talk about more of how they work. So each of the enzyme has an active site. Active site is where it will bind to the substrate, and that will be unique for each of the enzyme due to the variations in the amino acid residue, into how they're positioned in the 3D positions, or how the amino acid is being arranged. So we call that a sequence of amino acid, how they form the structural property. All of that will create a very microenvironment that is unique for each individual enzyme that will be very specific for the substrate. And because of this right here, that will cause the enzyme to have a certain specificity, and it's very specific. Of course, there's always some exceptions, so there's some flexibility that comes into being. So originally, we thought enzyme and substrate fit together as a lock and key. So we call them a lock and key model. It's like a really nudgely fit in there in one steps. However, recently we have more evidence suggesting that an induced fit model is more of what's really going on. As you can see here, this round part, but this part right here is not really round. It has a different shape. But upon binding of the substrate, the enzyme changes shape to fit this substrate better and to catalyze the reaction. Thus, it's called the word induced fit. Induced fit here means that the fitting of the substrate will induce the enzyme to change the shape and fit the substrate much better. And once you have enzyme and substrate bind together, you will have an enzyme-substrate complex, and that complex is where the reactions will be catalyzed or will occur. So enzyme will promote chemical reactions in a variety of ways. The first way they can do it is by putting it in the optimal orientations. They will orient the atoms and bonds of one molecule and put it in a way that will be in the correct regions or in the appropriate regions of the other molecules that facilitate the breaking and forming bonds between two molecules, and that's how they speed up chemical reactions and lower activation energy. Or two, they can create an optimal microenvironment where it make it a little bit more acidic or a little bit more nonpolar environment for the substrate to react. Because normally to exist within a aqueous solution is not always the best environment for any reaction to occur. They differ slightly, and enzyme provide that shelter, that optimal microenvironment that the substrate can react. The third way that enzyme can promote reaction is that they can contort or they can alter the substrate molecules in a way that facilitate bond breaking. They can put a strain on the bond, cause them to break easier, or they can put them closer together, that's closer to orientations. The point here is they makes the bond closer to these transition state, and the transition state is the higher energy state within reactions. And once we reach that state, it will go downhill and cause the reaction to occur. Lastly, the last way that enzyme can promote reaction is they will actually take parts in the reaction as well. So certain amino acid residue from the enzyme can interact with certain ions or chemical group from the substrate molecules. They can actually form covalent bond temporarily. They can form ionic bond. All of those bonds will allow for the reaction to occur much better. The one important thing is, despite doing all of these actions, enzymes do not change at the end of the reaction. They will return to its original stage. They will be unchanged when the reaction is completed. Of course, it will change during the reactions. That's very possible, as you can see in this number 4 right here. But at the end, when the reaction is completed, enzyme will return to its original state. Certain enzymes do not work alone. They require others non-protein helper molecules. We can call them a cofactors or a coenzymes. So cofactors are inorganic ions that participate in the enzyme molecule. They can be iron ions, they can be magnesium ion, they can be zinc. The second helper molecules, we can call them the coenzyme, because they are actually organic molecules that can help with the entire speeding up chemical reaction process. And they are actually requires for certain enzymatic reactions. The most notable example that we will see are vitamins. Vitamins are actually, they can act directly as a coenzyme, or they can act as a precursor to a coenzymes. So you have your vitamin A, your vitamin B, your vitamin C. There's more that you can see from the textbook, but these are the pictures. So these cofactors, or inorganic ions, or coenzyme, or organics helper molecules, they are required for certain reactions to allow enzyme to function better, or even it is a necessary component for that particular enzyme to make the reaction occur much faster. Enzyme is a process where it needs to be controlled because um, enzyme can be if it acts without any directions or if it acts all the time, it's almost a waste of energy. For example, there's an enzyme called lactase that can break down lactose from milk. Only when we drink milk or have lactose inside our body we will need lactose. If we don't have that, we don't need the enzyme to be there to facilitate the reactions. So there'll be other molecules that come into the pictures or come into the reaction to inhibit that certain enzyme functions. So we categorize inhibitor into two classes. We have a competitive inhibitor and we have an allosteric inhibitor. So the competitive inhibitor will directly bind to the active site that will directly compete with the substrate and preventing the substrate from binding to the enzymes. So that's competitive inhibitor. Whereas the allosteric inhibitor, this word allo here means other, allosteric meaning other site, or so the inhibitor will actually binding to the site that is not the active site, as you can see in the pictures right here. This is the active site, that's the active site. This inhibitor is an allosteric inhibitor. It will bind to that another site on the enzyme, and that will inhibit, it will change the shapes of the active site, or what we call changes the enzyme conformational shapes or conformations, and that will not allow the substrate to bind anymore. So that's called inhibitions. You can also have allosteric activation as well, whereas you have that activator bind to an allosteric site. So that's a site that is not the active site. That will cause the enzyme to change its conformations, and that will be able to bind to the active site again. So again, it's not only inhibitions. This regulation can be either as an inhibitor or inhibitions, or it can be an activation as well. So this is a little bit more about enzyme kinetics. So normally, on this graph, it's showing this normal enzyme actions right here based on its substrate concentrations. So on the y-axis, you have reaction rate, which is how fast the reaction occurs. And as you can see, as you increasing the substrate concentration, your enzyme will act the rate will occur much faster until it reaches a certain point. So this max rate occurs is because you only have a limited amount of enzyme. Up to a certain point, no matter how many more substrates you are adding, you will not have enough enzyme to perform all the work. Thus, you will have where it will plateau off into a maximum rate. The competitive inhibitor will block the substrate concentrations, thus it will dampen that rate of the reactions. However, when you add enough substrate, you will eventually be able to reach this max rate again. So the competitive inhibitor will only affect the initial rate, as you can see here from this purple line become the green line. The rate actually decreases along the way, but it will eventually reach the maximum rate. So that's when the substrate will outcompete the competitive inhibitor, and this will behave normally as what's happening without the inhibitor. The non-competitive inhibitor do not have to compete with the substrate. So not only they will affect the initial rate, they will also affect the maximum rate as well, because they do not affect the substrate. So it doesn't matter how many substrates are there, they will be able to work regardless of the substrate present. So at a certain point, they will also have plateauing off. So this non-competitive inhibitor line will be different from the normal enzyme in a term of it will lower the maximum rate of the reactions. Then lastly, throughout these inhibitors and activators, our body can actually put this in a feedback loop or a feedback inhibitions, where the end product of a pathway can potentially go back and inhibit one of the enzyme within the pathway, it's usually will be the first enzyme within the process. As you can see in this figures right here, this enzyme will facilitate that substrate, which is this green triangle, and convert that into an intermediate substrate A, this rectangular A, purple rectangles, and enzyme 2 will convert that into this irregular shape, blue shape, and enzyme 3 will convert that into the orange square right here. So all of this will make that orange square at the end. And when this is the end product, and that end product will actually go back to enzyme number 1, become an allosteric inhibitor, and will prevent enzyme 1 from catalyzing more substrate. So that will actually shut down the entire process because you no longer have the first step being initiated. So that's generally how feedback inhibition works. And this is developed through evolutions because we want to conserve energy. We want to have the ability to turn on and off pathway at will so that we can conserve energy and allow for maximum efficiency within our cells. Another example that is familiar to you is an ATP. ATP is actually an allosteric inhibitor for a certain enzyme that is involving in sugar catabolic breakdown. That makes sense. When you have a lot of ATP, you don't need to break down sugar anymore. If you keep breaking down sugar, you actually have too much ATP. ATP can actually go through spontaneous dissociation, like what we talked about before. It will be just a wasteful process to produce more ATP at that point. Then ADP, when you have high level of ADP, that signals that you don't have a lot of ATP, meaning you don't have a lot of energy. So ADP can actually act as a positive allosteric regulator or an activator for the enzyme that makes more ATP. So ADPs, high ADPs means that it has the cells has low ATP, and that can trigger ATP productions. And that concludes our chapter for today. I will see you next time in chapter 7.